Very warm welcome to everyone. The topic for today we made up together with the colleagues is about Luxturna, the first and so far only approved gene therapy for retina dystrophies, and we will split the topic between four of us who will talk today, and it's my pleasure to introduce all four speakers to you. We will start with Professor Bart Leroy, who is a professor for ophthalmology in the Ghent University with a large expertise in ophthalmic genetics, visual electrophysiology, and he is also heading the ophthalmology department. Bart Leroy has been a PI for many years for treatment trials, and he will introduce us to the background of how Luxturna came to approval and also the experiences from the currently running post-marketing observation trials. So Bart, thank you very much. I would like to give the floor to you, and we are very much looking forward to your presentation.
So thank you very much all, and it's such that I want to try and help cater for all of you. So certain things I'll be introducing will be too easy for certain people, others may find it too difficult. I'll try and cater for everyone. So as a clinician scientist, I was involved in the development of what is Luxturna. These are my financial disclosures. I work with a lot of companies. They all give money to my research department and not to myself. So when we think about what the eye does, the eye translates light into electrical signals, and it does so by using the human retina. The retina can be seen as essentially four different layers of cells: the pigment cells, the photoreceptor cells, the bipolar cells, and the ganglion cells, the latter making the optic nerve, which is the cable running to the brain. So the photoreceptors are of two types, the cones and the rods, and obviously the pigment epithelium has a very intimate relationship with both of these. So this is basically a functional unit. You cannot see the pigment cells separately from the photoreceptor cells. They work together intensely. Rods translate light into electricity as soon as there's very little light. The cones do that during the daytime, rods during nighttime. So this is, for example, a rod outer segment, and this is where it really happens. I always see this as a stack of discs. Often I name them a stack of pizza calzones. And in the pizza calzones, the closed pizzas, float in the dough of the pizza calzones, float quite a few proteins that are all together involved in what we call the phototransduction cascade. A phototransduction cascade that actually translates light into electricity by using vitamin A, and vitamin A is intimately bound to the orange protein that is visible on this slide. The vitamin A is basically used in that process when an electrical signal is being generated by the closure of a channel in the cell membrane. So we have to recycle vitamin A continuously, and we do so for the large majority of the vitamin A, and that is done through the retinoid cycle. The retinoid cycle is throwing vitamin A that is used in its all trans isomer out of the photoreceptor into the interphotoreceptor matrix. It's then being picked up in the retinal pigment epithelium cell, and then gets recycled and thrown back for it to go once again. And basically, if we look at the inherited retinal diseases that we call IRDs, most of them involve the cones, the rods, or the pigment epithelial cells, that is, outer retinal cells. So Leber congenital amaurosis is one of those IRDs. It was described by Theodor Leber, a German gentleman who described quite a few conditions, and he described what is now known as LCA as a condition that led to the retina not translating light into electrical signals. So children from very early on in life do not seem to capture much light. They don't have visual interaction with their parents. And when an ERG is measured early on in life, we can't measure any electrical activity coming from rods or cones. So 24 genes and counting now, they are responsible for about 70% of the known patients. And one of them is RPE65. You can see where all these different proteins encoded for by these genes work, and they do very different jobs. But the final common pathway is that the retina doesn't translate light into electrical signals. One of those genes, RPE65, encodes for a protein that is essential in that retinoid cycle, which is the recycling of vitamin A into from its inactive to its active form again. As we continuously translate light into electrical signals, we also continuously need to recycle most of our retinal, which is the vitamin A. So Professor Christian Hamel was the first to discover the gene in 1994, and it was also Christian Hamel and Andreas Gall who in parallel described in 1997 that this was also a basis for the disease. So when we look at the recessive retinitis pigmentosa, or the retinal dystrophy, or the Leber congenital amaurosis, that is due to the fact that there are two mutations, one in each copy of the RPE65 gene, then we see that initially the retina looks fairly normal. Very typical is that with a blue light autofluorescence technique that we use to see lipofuscin, a degenerate material that is continuously produced in our retinas, we see very little on our images. Later on, the retina evolves to a situation where it doesn't look any different from classic retinitis pigmentosa. But we do have some time to interact with the retina and make sure that the retina that is initially not completely abnormal starts working again. And so there's a little bit of a window of time to work. So getting from animal models to human treatment is the next part that I want to zoom into. So when we want to add a novel copy of RPE65 in this case, we can use viruses that transport, basically like a little van that does deliveries at home. We can use viruses of the adeno-associated type to bring to a target cell a novel copy of a gene that nature has given that cell two bad copies of. And so here we use adeno-associated virus type 2, driven by a chicken beta-actin promoter, and it contains one copy of the RPE65 cDNA, which is the short version of the gene, and we inject it subretinally. So we do a vitrectomy, get the gel out, put some liquid in. Why the vitrectomy? Up to now, we think that if we work in the gel, the vitreous gel, we can pull too much on the retina. We may actually provoke retinal tears. So there are also gene therapies that inject into the vitreous directly. But for this talk, we are speaking about subretinal injections. So what does adeno-associated virus basically do? It enters into the target cell. It releases the payload into the nucleus of the target cell, and the cDNA forms an episome there. It doesn't normally insert in the genome, which is good because it then cannot induce mutations. The post-mitotic nature of the RPE cells, the fact that the RPE cells don't divide anymore, allows for a continuous presence of that gene, because in dividing cells, one would lose an episomal gene. So when we worked towards getting this treatment, Voretigene Parvovirc, the adeno-associated virus that transports an RPE65 gene, we basically were hacking a path through the jungle. There was no road available for us. We were the first to do this. So the first example is this video is illustrating this, is that in dog models, naturally occurring dog models, treated with gene therapy through subretinal injections, we saw that there was an improvement of function. What you see here is a large dog who is normal. We see two little twins. One is treated, the one that is very active, and the other one, for those people who can't see the screen, I'll describe it. One of the two younger dogs is actually not able to partake in playfulness, whereas the treated dog with gene therapy is actually acting as normally as the larger dog who was not diseased. So then we moved from dog models into humans. And what you can see here is Lancelot, the dog that was actually taken to the House of, to Congress, to the House of Representatives and the Senate by the people from Philadelphia. And he's reading the paper that his paper was published in Nature Genetics, and he's reading this. His Lancelot is now dead, but he was a very nice dog. In 2008, three parallel publications from Robin Ali in London, Jean Bennett in Philadelphia, and Bill Houseworth in Philadelphia and Gainesville published papers that saw phase 1 trials show that this was an efficient treatment, that you could change retinal function. Eventually, there were 8 different clinical trials. Some were related to others, but a total of 98 patients were treated in the trials between 2007 and 2009, and they all differed in what type of subjects were included, the dosage used, etc., etc. But in general, it was considered safe and sound. So we in Philadelphia basically published phase 1 and a phase 2, phase 1 and a phase 1 follow-on study. The follow-on study was where 11 of the 12 initial patients were treated in their second eye as they were older, to see whether bilaterally a gain of function could be had. So the development history of what is now known as Veretigene neparvovec, also known as Luxturna, is a long one. If you look at 1997, the gene described as being at the basis of the condition, and 2017 as the yes from FDA, that's 20 years. So we did a phase 3 trial design, or a phase 3 trial, the design of which you can see on this slide, and I'm not going to dwell too much on the details, just to say that we had 29 patients partake in the study. The phase 3 study is a pivotal study. It's basically what helps something to be brought either to market or not to be brought to market if it's inefficient or too dangerous. And so we had 20 patients initially treated, and we had 9 patients who waited for 1 year to see whether naturally they would evolve towards an improvement. Obviously, they didn't, and so after a year, they were part of what we call the delayed intervention group. The original intervention group was the first. We had to come up, based on our studies, the phase 1 and the phase 1 add-on study, with something that we now know as the multi-luminance mobility test. Why? Because we didn't see that there was significant improvement of best corrected visual acuity. For some reason, the cones in the fovea didn't really improve in function, but what was abundantly clear from the phase 1 and phase 1 add-on was that the retinal sensitivity shot up. And how to express that better than using a multi-luminance mobility test, where 7 light levels were used, and people were asked to navigate from point A to point B through an obstacle course. Time was tested or measured, and also it was measured how many times people bumped into objects on the navigation course. So we also did full field light sensitivity, which is the full field light sensitivity threshold test, or FST, and we obviously also measured BCVA. So here you see a depiction of what is the multi-luminance mobility test, and basically what we saw was that the sensitivity shot up initially after the treatment quite rapidly. After 1 to 2 weeks, we saw mobility being much better than before. after treatment for the original intervention group. And then after 1 year, the same happened to people from the delayed intervention group. And nothing better than to show a video. So for those who can't see the video, what you see on the left is basically the same patient. She's a Belgian patient I took to Philadelphia. On the left-hand side, you see her prior to treatment, move through the navigation course, the obstacle course. She doesn't do a good job. She actually fails the test. And at the right-hand side, you can see that after 1 year, the patient basically moves through very rapidly, and she's already done whilst she's still struggling prior to the treatment on the left-hand side. So we won't dwell on that. I'm also not going to dwell on the fact that the best corrected visual acuity didn't really statistically improve. It did in some, it certainly didn't do in others. What did improve, though, was actually the mean Goldman visual fields with the object 34E of Goldman. And we also saw that the mean macular threshold as measured with an automated Humphrey static visual field did increase as well. Safety was also very much acceptable in the sense that we saw some increases in intraocular pressure. We saw a cataract, we saw retinal tears, we saw some deposits, and that's interesting because I'll come back to that. There were no serious adverse events that were really related to either the drug or the intervention, and so it was deemed safe to do so. And we saw that there was a significant improvement in MLMT, multiluminance mobility test score, FST, and visual fields at year 1 in the delayed intervention subjects, as was initially there in the original cohort. And we basically used the results from this trial to bring about an introduction to market because it was approved in 2017 by FDA, EMA followed on in 2018, and so it was introduced to the both US and EU markets. So we basically have a need for some retinal cells to be there as evaluated by an expert in IRDs. We do have to have at least light perception vision, and it is desirable to also have one measurement, an additional measurement of visual function. So for example, FST, and then you can treat with this drug. The drug basically is nothing else than 300 microliters, which is a big drop of liquid with 150 billion viruses, each one of them containing an RP65 gene that is subretinally injected. So how long does the effect last? And that's the next part of my talk, and it'll be basically the end of my talk. I'm going to go through 2 studies that you will see, but where are we with the results at long term? So I can definitely show you here the 5 and 4 years, so 5 for original intervention, 4 for delayed intervention from the American phase 3 study. And you can see a continuation of the gains made both in the Goldmans and the FSTs. And what we also have learned since the introduction to market is basically that there is a problem with chorioretinal atrophy. There is inflammation to be seen, and we still have questions regarding the effect and how long it lasts. And the first thing I'm going to quickly mention, I'm sure that Professor Stingl is going to zoom into that a little bit more, but the group of Aaron Nagel, our friend from Children's Hospital of Los Angeles, published in 2022 a series of patients where they said, well, at least in a subset of our patients, we did see some chorioretinal atrophy. And that really focused our minds. And personally, I think that there are 3 types of chorioretinal atrophy. One is chorioretinal atrophy seen at the injection sites, which is normal. Then you have chorioretinal atrophy within the treatment area. That's worrisome. And then treatment beyond the treatment area, which is potentially at least due to normal evolution of disease. So for example, here what you see is an unpublished series of pictures that I share with you. This is our patient who was the first child ever to be treated with the condition, with the drug for the condition RP65-IRD. And on the left top-hand side, you see a fairly good fundus. You see that he does show some chorioretinal atrophy beyond the treatment area. And then progressively, you see also some atrophy in the treatment area after 6 years and 10 years. This is seriously visible. What we do know also is that there is a need to better control the inflammation that highly likely is provoked by the AAV2 that we use as a vector to bring to the target cells a novel copy. And there are now multiple papers. I'm not going to dwell on this. For now, you will hear from my colleague Katarina. The data from our center is that we've treated now 32 eyes from 17 patients, and we've seen that there is some atrophy in the area of treatment, and that is mostly seen in the injection site, but some of the patients do show some atrophy also in the treatment area. And we are currently investigating what to do. So I can show you this young girl when she was treated at age 5-6 years. And what you can see on the four photographs is OCTs. And you can see that there's chorioretinal atrophy happening just temporal in this left eye, temporal of the optic disc. And this is in keeping with the presence of subretinal deposits. We now think that they were subretinal inflammatory cells that we failed to identify at that time. And I'm still beating my head over it. I should have seen this. I should probably have been more aggressive in treating her. However, she doesn't have any loss of function. So what about the PERCEIVE study? And those are the two last things I'm going to show you. The PERCEIVE study is all the patients treated in the EU have to be entered into a phase 4 study that EMA asked us to do. And what have we learned from that? And then what have we learned from a similar study that is happening currently in the US? This study has, I'm going to not dwell too much on it, but 527 eyes have received Veretugene Neparvovec at the time of the data cutoff that we are presenting here. And so basically, the large majority received a full volume. And what I'm going to mostly focus on in these 291 patients who were enrolled is what the results are regarding function, but also regarding the chorioretinal atrophy. And so these patients are coming from all over Europe. And if we look at the chorioretinal atrophy on the right-hand side mentioned in the table, the study mentions that nearly 30%, that is 29.6% of the patients, or 27.3% of the eyes, actually show some level of chorioretinal atrophy. This is a mix, an amalgam of all types of chorioretinal atrophy that is both at the level of the injection site and also within the treatment area. And so we need to split those up to better understand. That's my personal opinion. But chorioretinal atrophy is certainly something that deserves more attention. We also had some other issues like glaucoma and so on, but I think in general it was deemed safe. Interestingly, when we look at the total group of patients, the FST showed a considerable improvement of the retinal sensitivity in virtually all the patients, whereas the best corrected visual acuity didn't significantly change, and therefore also confirmed the findings of the phase 3 study. And then if you look at FST, that is the retinal sensitivity by presence of chorioretinal atrophy, what you see is two groups divided up by the presence of or the not presence of chorioretinal atrophy, and you see that actually the group with chorioretinal atrophy is doing slightly better. At least you can say that actually there's no change in function when you have chorioretinal atrophy. The gene therapy seems to increase your retinal sensitivity significantly. That's what it means. I'm not going to dwell on these. Let me just quickly to end show you the safety and durability in the at 8 and 9 years, which we recently presented at the American Society for Retinal Surgeons in Stockholm. So I don't have to dwell on this. What I can show is that the mean bilateral multiluminance mobility test change score didn't change much over time up to 8 and 9 years, depending on original intervention, delayed intervention. So there is a little bit of a peak that we saw, but that has to do with the fact that there are very few patients. What I'm going to show you here is a video, and this video on both sides. On the left side, you see a patient move on the left-hand side through before treatment, failing the course. After 1 year, he passes. You see the same video again. On the right-hand side, there's also 2 videos on the right-hand side. On the left video on the right-hand side, you see the 1 year, and you still see that after 9 years, the person passes the test again. And so that shows you that, for example, in this patient, and many of the patients show that, they have a result that is steady over time. So there are improvements in light sensitivity and visual fields up to 8 and 9 years post Luxturna administration. The visual acuity also remains stable. It's not going down significantly over time. It doesn't improve over time. And then the cumulative ocular adverse events show obviously cataracts. There was in this evaluation no specific attention for chorioretinal atrophy. That is remarkable. We are currently looking at all the data because obviously we are going to find chorioretinal atrophy over time also in this group. And so basically, what have we learned from this? Faricogene neparvovec works in RPE65 IRD. It doesn't normalize visual function. It improves with now data available up to 9 years from the phase 3 study. I can tell you from the phase 1 and phase 1 add-on study that the function still is pretty okay after 14 years. I haven't specifically mentioned that, but 2 patients I personally follow, they still have significant improvement of function after 14 and 13 years. The inflammation is incompletely understood and requires further study, as does chorioretinal atrophy. And so the need for genotyping, and you'll hear from my colleague and friend Elfride de Baere, as a molecular geneticist, she knows it better than any one of us. There is an enormous need for genotyping if we are to bring gene therapies to patients. We are now beyond 8.2 billion people in the world, and that means we're nearly at 3.3 million people with IRDs. We want to help our patients. Patients want to be helped. The industry wants to make a little bit of money doing these developments, and the regulatory bodies obviously want reliable drugs. So there is a big need for us to get together. We are doing that currently. The future, I think, is bright. It's a novel field. It's falling flat on your face, standing back up again, going further, and taking quite a few hurdles. But I think personally that we'll get there in the end. The Ghent Ocular Genetics team is much more than just Bart Leroy. I am proud to be the leader of a group of incredible people. Elfride is in the middle here, but obviously she has her own team. But I'm proud to work with this, and thank you very much for your attention, and I'll stop here.
So Bart, thank you very much for this excellent overview. We will go on with the second presentation, which will be given by myself. I'm ophthalmologist working at the University-EYE Hospital in Tübingen in Germany, and I'm specialized in IRD diseases, taking care of the Center for Rare Eye Diseases, and in our group we are performing also several clinical trials, gene therapeutic, but also different. And I would like to share with you the thoughts or the lessons learned that we were able to collect in the years after approval. I have written here, what have you learned in 5 years? Well, basically it should be 7 years now, but let's stick to 6 years because the 2025 just has begun, and we did not treat patients in the first year usually due to insurance issues. So what about the medical ophthalmological things that we learned? I will start with the slide you have seen already, or the basic, and I don't want to repeat myself from what my colleague Bart explained very well, but just to remind you, the gene RPE65 is an enzyme sitting in the pigment epithelium cell and is very important for the regeneration of the rod visual cycle, for rhodopsin, and in its absence, the patients suffer usually an early childhood retinitis pigmentosa, or better say early onset retinal dystrophy, in the extreme case very high visual impairment in the form of LCA. Typical is this low fundus autofluorescence, as you heard already. But speaking about the rods, the first lessons that we kind of learned in treating and following up patients and really wanting to understand the treatment effect is that you have to rethink a little bit the diagnostics that you use in the normal IRD routine with follow-ups. Usually these are focused on cone diagnostics because cones are those cells that matter in the daily life, visual acuity, visual field, and so on. But here, treating rods, you have to really be aware that you might not be able to catch the effect if you stick to the original diagnostic that you do for follow-ups, but you have to focus on rods. And of course, as Bart already mentions, there are patients who really show very nice treatment effect on cones, but they are not all of them. And if you want to understand what happens in the retina after this treatment, then you have to focus on rods. So this is just an example, if you maybe for you to better understand what I mean, because this is a patient who actually had a very good treatment effect. And this is a young female with a baseline visual field, here concentrically reduced to the middle, and BCVA in German decimal way expressed 0.16. And then after treatment, although there was really a huge and very good treatment effect, if you use these two common measures, you maybe miss the actual happiness treatment of the patients. So the visual field stayed more or less the same, and the BCVA also. So what is the answer? The answer is, of course, what we have learned already. For example, dark adapted threshold testing, also chromatic testing, not only with white lights, as was performed with in the approval trial. And this is an example of FST readout that is able to capture very well dark adapted improved sensitivity. That means improved rod function. Here depicted over the time, and let's have a laser pointer here, and depicted over time from baseline with quite bad values here at the beginning, and then improving for red light and blue light, but also for the white light. Just to remind you, white light was used in the trial, but a combination of red and blue can give you also a very good demonstration of this difference between the two thresholds, showing here a clear rod functionality. And this is what we have learned over the time following up the patients. And this diagram shows you only the blue color, which represents rods quite well. And what you see here is again the time scale, but very interestingly, we are able usually to see a very profound treatment effect 2 weeks, sometimes 4 weeks after the treatment. So the effect is pretty fast, but then over the time, the effect a little bit lowers down, and after the 3rd month stabilizes with some decent oscillations over the time. So this is quite interesting, and I think judging the treatment effect before 3 months might not be a stable value for the treatment effect, but already at month 1 you see a quite clear change. So let's show you some individual cases in order to go a little bit deeper into what we have learned. This is an example of a subretinal bleb injection, a little bit irregular, but it's not that important. Here the treatment was applied. I was again a young patient, a teenager, and if we want to have a look at the rescue of the rods, we can use FST, but we also might be interested how the rods did improve exactly in the area where the patient has been treated. And for this, an excellent device is, of course, the dark adapted perimetry. This is one commercially available device. I think there are currently 2 of them, without doing any advertisement. And when you do this, then you see here on this slide in the beginning, basically no recognition in the dark adapted state for the patients. On the right side is a normal, like what I would perform, or probably any one of you. And in the middle is then patient treated here 3 months after the gene therapy with vorticine Parweg, seeing quite well that the best improvement is in the treated area, but spreading a little bit also across the fields of the bleb. And here you see an improvement of up to 40 decibels, which is really huge. And normally in young patients, we are very happy about an improvement about 20 decibels, which is rather frequent. So if you do a little bit math, then you know that 40 decibel means that this patient on this spot is able to perceive 10,000 times less light levels than before treatment. And that is, of course, very big improvement in the daily life, although the visual acuity does not change. And a small video, I'm not sure if it works, and if not, then we just skip it. And it shows you a 10-year-old girl who, and this is the second one, the first one was a friend of her, who is able to jump in the dark garden over this fence. And this is, of course, something that she would never be able to do without the treatment. Family is extremely happy. Okay. So what do other patients report? So patients report usually improvement of vision in the lower luminances. There is an example of being able to perceive a cup in a better way. A child is able to look at him or herself in the mirror in the night, and so on. Statistically, there is usually a report of brighter vision in the most of the patients and improvement vision in the darkness. Some of them basically due to this improved brightness of perceived luminances can see a contrast better and improve their orientation. But there is a substantial amount of patients who again perceive the higher luminances as too blinding, and some of them also suffer from headache after the surgery. I have to say, I do not know if there is a causal connection. This was just recorded in our patient population, and this headache usually disappears during months after treatment. So not every one of the patients does have this very exciting rod rescue function, and those who don't are usually patients who are older, and we see here some kind of age-related effect of the rod improvement. So FST blue sensitivity direction down is good, and the massive improvements are seen rather in young age and then around 30, but this can really vary individually. The gain is not measurable, although we do believe that the progression could have been stopped. There is maybe, the sentence is maybe not that correct because the effects are not age-related, but they are related to something that is related to age, and this is availability of remaining tissue. So as you have heard in the first presentation, it does matter if I have cells available that can be treated, and such a patient definitely will have a good expectation for the treatment benefit, and this patient in a late stage probably will not. I would like to spend some words on cones again, and we have heard that there were patients who improved BCVA, there were patients who did not improve, and yes, there is an effect on cones, but it's not measurable in most of the patients. Our data show more or less with some oscillation a stable BCVA, and this is just an interim result. It's a little bit older already, and in another publication where we examined pupilographically cones in a cone-specific protocol, you can see that a local pupillography, if this is the baseline pupil response over after a cone stimulus, and the blue one represents the normal response, that the response measured by pupillography can be even normalized. So there is a direct effect on cones, although you cannot rely on this when judging the treatment response. What about the retinal imaging? Well, this is quite interesting, and we know that autofluorescence is very difficult in these patients. In some of them, you just get a noisy noise picture. But interestingly, in many patients, although this is not 100% true, but really in many patients we see with the improvement of the retinal ability to process or to have the enzyme RP65, basically also a better imaginability of the patients. And this is one example. And signal can be improved after treatment, but again, it's not guaranteed, although it's seen in many patients indeed. OCT, it's rather rare that you can measure the effects in OCT, but there are single cases like this one where you can truly happily observe the OCT findings over the time. And if you see here, this quite young girl has had a very bad foveal. She had this pattern of rather central degeneration, and then a pericentral a bit better, and then again peripheral atrophy. She had a disturbed foveal structure before treatment, and then already 2 weeks after, and also in the following weeks and month, you can see a little bit different pattern of kind of starting again a restructuring of the foveal region with measurable, this is probably external limiting membrane, or maybe EZ zone. You don't know if the other line is missing, but it correlates to a very slight improvement in the visual acuity, or you maybe say stable vision. The OCT is useful in monitoring efficacy, but also useful in monitoring safety. And what you see on this right-hand side is a patient who suffered a loss of the retinal thickness. We have had only one case like this among our, I think, around 25 patients currently. That is a known adverse effect, retinal thinning. You cannot predict this. There will no mistake in surgery here. But again, a decay of a local visual function in this tint area is then explained by the morphological findings. And here I come back to the safety issues that have been already mentioned a little bit in the first presentation, and those are inflammatory reactions. Inflammatory reactions, yes, they do exist. I have to admit we didn't have any such deposits, but this is of course not influenceable. Prior to the surgery, patients do get prednisolone around the treatment, and especially children, and this has been published also by the Los Angeles group, children do tend to or are prone to create this subretinal deposit, which resolve over time, at least what has been reported also by other colleagues, sometimes with additional cortisone treatment. Okay. And the next topic are the already mentioned chorioretinal atrophies, which is a phenomenon that has gained our attention after approval only. And you see here example of these two groups. A patient like this, she is a patient who over 2 years does not show any evidence of these atrophies, but here is an example of these atrophies that you can see mostly around the large vessels with this patchy pattern with a confluence over months or years, and they usually start to develop at month 3. There has been this publication from the Los Angeles group as the first one, and you see here this atrophy quite well evolving in the treatment area. And what we can see is these atrophies are of different kind, as you already heard. Some of them are really arising only from the retinotomy. They grow a bit, but they are not unexpected. But what I am talking now here about are these not retinotomy related, mostly connected to the treatment area, but also partially beyond loss of cells. So yes, there can be natural progression, and you have seen a patient from Bart where it could have been also natural progression. We sometimes do not know. Then there is a surgery-related risk, but this would not explain why there is a substantial group of patients who do not develop these atrophies. And again, some of these atrophies do not evolve in the area where the bleb has been detached. Immunological factors are, of course, important, but again, immunology is also a reaction to a cell death, so we can find some immunological reaction in these areas. We do not know, is this the cause or is this what comes after? And also all patients do get prednisolone, so this would not explain why some of them develop these atrophies, some of them not. We have had a look into the data with the colleagues in Los Angeles, and in a way to find out which factor is a kind of prediction factors or risk factors for this development. And I would like to explain you this fourth possible explanation, which has to do something with retinal metabolism. So this is the table that we gained with colleagues in Los Angeles with altogether 71 eyes, and we checked, this is just an example, we checked several factors of the patients and looked what does make the significant difference. The most significant difference, what we have seen here, was the initial improvement in FST, and we evaluated these patients really only for these mid-peripheral atrophies, not for those starting from the retinotomy. So you see that there is a highly significant difference of the initial FST between patients who do get atrophy and patients who do not get atrophy. And those ones who developed atrophy are those ones who benefit from the treatment in much higher way than those who do not have atrophy. If you want to visualize this over the age distribution, then you see that the patients who are prone to develop these atrophies are mostly these young teenagers or young adults who have quite a good treatment effect. And here comes the hypothesis that might explain this phenomenon quite well. Now you can imagine that there is a kind of substantial degeneration in the retina already at the treatment of a teenager, and now suddenly you increase the efficacy of the rods by 20-40 decibels. Rods and cones, as you know, are the most metabolically active cells in our whole body, and if they increase their activity in such a large amount, the degenerated retina might not be able to cover the metabolic needs. And this is then a kind of reaction of the tissue that reacts with cell loss as a price for the efficacy. This also explains why patients who are rather not that good in their rod rescue, they are rather not developing efficacy. Here is a little bit mixed because, of course, every patient is different. And what is very interesting is this young cohort. These are preschool children who are excellent in their expectations of treatment effect. They have a good dark adapted gain, but they do not develop atrophies. And you can realize that this cohort has a retinal state usually that did not pass kind of level of no return of the degeneration. So if you here recover rods, it probably doesn't have this metabolic effect that results in subsequent cell death. So anyway, early treatment seems more beneficial, and this analysis only supports the fact the earlier treatment, the better. And in my last slide, okay, this is the second to last slide. This is just a visualization. We ophthalmologists sometimes think when we just replace only one part of a broken car that we might get a Rolls-Royce, but we don't. In fact, if you just replace one enzyme in a degenerated tissue, it can lead to even more problems and will not work. And this might be that what happens in the regions of choroidal atrophy. And now in the really last slide, I would like to explain you last thing that we learn based on our cohort and evaluations, and I will walk you through this slide because it might not be that easy to understand. So these are examples of two patients. Number one, first line, number two, second line. We have here these images of the chorioretinal atrophy growth, which starts usually at 3 months. So you see here starting and evolving in the large areas at 12 months. And we had a look what happens in these areas functionally in order to say, is there a prediction factors of this metabolic imbalance to that what happens static with month 3. And what we have seen is a very interesting pattern, because here is the dark adapted perimetry. And red areas are those who improved very good. And as you see, here have been rods. They have, would you please switch your, yes, thank you. Here have been rods that improved in their sensitivity. So the area had rods, but they just got better. And those are the areas that do not develop atrophies. And by the way, those are the areas that probably take the effect for long years, and we know that effect lasts for 5 years now. And what is, this is another examination, is a pupillographic test, which evaluates the local function, indirectly, of course. And what you see here is a gain of number of rods that has become active with the treatment. And those areas usually correspond exactly to the spots that are prone to develop atrophy. So the interpretation of this slide showing our recent paper is that exactly those spots of the retina that have lost a lot of cells before but regained the cells after the treatment, they enter into kind of metabolic disbalance, if I might say, and are prone to develop atrophies. But in contrary, those areas that still had cells but the cells only improved, those are the areas that can take the effect for the subsequent years, and we can measure that, of course, in FST as well, but not resolved on the retinal level. So I think these are lessons that, of course, not able to be evaluated before approval, and we are very happy that we have this opportunity to study new methods. And I would also like to thank the whole team, because results shown are a teamwork, and a special thanks goes to Melanie, who takes care about all our Luxturna patients since 5 years. And I would like to thank you for your attention, and I'm of course available also later per email anytime. So thank you very much. I would really like to go to the next presentation of Elfride de Baere, who is a professor for genetics, also in Ghent University. She's heading the lab for medical genetics. I apologize, and is specialized not only in retinal dystrophies, but also other developmental eye diseases, and would share with us her experiences with the genetics and if anything can be improved. Thank you, Elfride. Please.
Thank you so much for the introduction. So I will follow up on what has been said about the clinics. I would like to zoom in on genetic testing and how to deal with the results of genetic testing. I'm working as a geneticist at the Ghent University Hospital, and I'm co-chairing the transversal working group together with Susanne Kool on genetic testing. I have no financial disclosures. And I would like to talk about three main topics. First of all, I would like to discuss what is today the standard of care in most European genetic testing laboratories for inherited retinal diseases, including RPE65. Then I would like to discuss the problem we are all facing, the so-called VUS problem. And then I would like to talk a bit about the future. First of all, how do we do genetic testing today in the clinic? So what I tell about RPE65 is also applicable to other inherited retinal diseases. And I'm trying to give a summary of what is the standard of care in genetic testing laboratories, in clinical genetic testing laboratories. First of all, we always start with the clinical diagnosis, and mostly this is done by expert ophthalmologists. Expert ophthalmologists or clinical geneticists see patients with unexplained retinal diseases and make a clinical diagnosis. They do a sample collection, and then we receive a request from a physician. And this can be the genetic testing that is being offered can be either single gene testing, although this is not the standard of care in most cases. This can be a targeted gene panel or exome sequencing based testing. Based on the clinical information on the outcome of genetic testing, a genetic report can be made, which is communicated with the referring physician. And then what is important is that there's a multidisciplinary interpretation of genetic testing results based on the outcome of genetic testing, the variants, but also the inheritance, clinical features matter, predictions, and literature, as I will illustrate. And then in some cases, the genetic testing result may result in reverse phenotyping and a re-evaluation of the clinical findings in the case of, in light of likely pathogenic variants. So mostly, no matter the test you do, you put a filter on it. This can be an exome-based testing, for instance. You can put an in silico filter on it. And I give here the example of a RetNet filter that can be put on an exome-based testing. But you also see examples of other filters that you can put on genetic testing data. In some cases, targeted gene panels are being done. For RPE65, this is mostly not the case. There, the standard of care is that you offer a broad panel for genetic testing. And then if you have a very particular genetic diagnosis, then single gene testing can be offered, as is the case for Stargardt disease. And then if we have done a genetic test, we look for single nucleotide variants, small indels, or copy number variants. So this is what should be done today in clinical setting. Here I give the example of the patients that underwent Luxturna at Ghent University Hospital, and you see that they comply all to the guidelines, what is needed regarding a genetic diagnosis. They all have two variants, so they are biallelic for RPE65 variants. You also see that the variants that have been classified are class 4 or class 5 variants, likely pathogenic or pathogenic variants, and in most cases, parental segregation has been done. In one case, this was not possible, but segregation has been done via the daughter. And then in two patients, there we didn't have access to material and to the parents, so segregation testing was not possible in these patients. So if we have done the standard of care, then we are confronted with many variants, and then we have to tackle the next problem. The next problem is the VUS problem. And what is a VUS? A VUS is a variant of unknown significance then the challenge is to determine pathogenicity of the variants that we find in RPE65. But if we do a broader panel, then we won't only find variants in RPE65, but also in other genes. And then we can classify variants in one of the 5 classes as prescribed by the American College of Medical Genetics and of Pathology. So we follow the recommendations published by these societies. And this is basis classification on family information, the nature of the variant, and different evidence level, such as population frequency, genotype-phenotype correlations, functional data, and also very important segregation data. So here you see a couple of publications that have been published by the American College of Medical Genetics. And here you see the standards that we follow in the clinic for variation interpretation and classification. And I would like to draw your attention to an important initiative from ERN-EYE. So we started with a sub-working group called REDVAR. And REDVAR is a multidisciplinary working group that is co-chaired by Bart Leroy and myself. So we have 4 times or 3 times a year, we have a meeting. And the focus is to discuss with a multidisciplinary team consisting of clinicians and also clinical molecular geneticists researchers to discuss RPE65 variants. So this was the initial aim of the REDVAR working group, but now we are also shifting to discussing variants and patients with variants in other actionable genes, and LRAD is an example. Then we also have webinars about genetic testing and variant classification. So if you're interested, then you can have a look at the replay of a webinar on this topic. I also would like to draw your attention to parallel initiatives. This is a mondial initiative by ClinGen, and ClinGen is a clinical genome resource oriented toward a better annotation of genes in the genome and variants in the genome. There is a ClinGen VSEP that is oriented toward curation of variants and also to specify the classification rules for genes implicated in LCA and in early onset IRD. And so now the rules have been specified by this VSEP, and several variants have already been curated following the FDA approved procedure that is established by ClinGen. So if you're interested, you can have a look at the ClinGen website and see which variants have been curated already and are curated and classified as pathogenic, likely pathogenic, and so on. And then there is another initiative from GenCC, and this is a consortium that is more oriented toward gene curation. In collaboration with ERN-EYE, we also have collaborated with EMQN, and EMQN is a European external quality assessment body for genetic testing. This is a pilot that you can see here, a pilot on inherited retinal diseases that was launched in 2023 in collaboration with ERN-EYE. Now the pilot will be replaced by a regular external quality assessment scheme, and the focus is initially was RPE65, but now also other actionable IRD genes are included in the external quality assessment scheme of EMQN. Here I will give a brief example of reclassification in a patient with RPE65 associated IRD. And the question was mainly about the classification of the first variant, which is a missense variant. For the second variant, the classification was a class 5 variant. So how did we do this? How did we tackle this? Here you see the different criteria, the different evidence criteria for which were used for the initial classification of the variant, which was a variant of unknown significance. Then, after discussion within REDVAR, we discussed genotype-phenotype correlations, and we agreed with the multidisciplinary team that the phenotype could be taken into account. So PP4 could be included, leading to a classification to likely pathogenic variant. And then And when we did a simulation, and we did a simulation with inclusion of functional evidence that this variant could be pathogenic, then we could reclassify the variant up to class 5 or a pathogenic variant. So the conclusion was that the first variant could be classified as a likely pathogenic variant. We could also show that this variant was in trans with the second variant. So we had a confirmed molecular diagnosis that could be reported. And the likely pathogenic and pathogenic variants could be reported. Class 3 variants are only reported in several cases. For instance, if they occur in trans with a pathogenic or likely pathogenic variant. If you have a homozygous FUS with very strong predictions or a de novo variant, then it can also be reported. Class 1 and class 2 variants are not reported. So the take-home message for how to tackle FUS is that based on clinical and familial information, different types of evidence, not to forget the clinical features, that genetic diagnosis can often be made. The predictions are not perfect, of course. Variant class does not always, it's not 100% certainty and doesn't always represent the severity of the disease. And also we have to be aware of the limitations of the test that we are offering. And that brings me to the last part. If we do what we can do nowadays in the clinical practice, we still have patients that are undiagnosed. We still have patients with missing heritability. With exome-based testing, this is around 40–50% in some cases. What are the reasons? There are limitations of exome-oriented genetic testing, and an obvious cause is non-coding variation. Another cause is gaps in the exome. Also, complex structural variants may be missed by the current mainstream approach. And also, we see an emerging role of complex alleles where we need to do a lot of segregation analysis before we know which variants are in cis and which variants are in trans. And this could be supported by new technologies. The complex and the cryptic structural variants are quite common in retinal diseases and could be tackled by using new technologies. We have been using short-read sequencing the last couple of years, and we are now evolving toward new technologies. Here we have a notorious example of a gap in the exome that also requires novel technologies, such as long-read sequencing. And this is important because this is a target for therapy, RPGR or 15. We are evolving from exome to genome testing. Genome testing involves one-size-fits-all test, where you sequence the full genome without enrichment. If your coverage is high enough, then you're confident that you can detect pathogenic variants at the nucleotide level, and also you won't miss structural variants. And also you won't miss variants in non-coding regions. But we are also shifting from genomics to multi-omics. One layer is not enough anymore. We also do need customized functional assays, for instance, for RPE65. In some cases, transcriptomics may help to explain variants, and then epigenomics will also be another layer that may be important in the future. So here you have a visual representation of how we shift from genomics to multi-omics, where we need an integrated approach. And I will give a brief example, not on RPE65, but on another example. For instance, this is here a deep intronic variant that was identified in OP1. We did predictions, and the predictions pointed towards the inclusion of a pseudoexon. And also a minigene assay in vitro showed the inclusion of a pseudoexon inclusion. And then we had the opportunity to do short-read RNA-seq on clinically accessible material showing the inclusion of the pseudoexon. And we also showed a difference between the in vitro and the prediction approach, and then the in vivo approach on accessible material from a patient. So this is, shows the added value of this additional layer on the genomics layer. Apart from this shift, we are also shifting from next to third generation sequencing. Here I show the difference between the output of short read sequencing, next generation or second generation sequencing, and long read sequencing. So you see that the read length increases, and this goes along with more information. This is the longer molecular weight DNA that you need for such approaches. We have two big players today for long read sequencing. We have the SMRT technology that is provided by PacBio, Pacific Biosciences, and then we have another long read technology that is nanopore sensing technology for the reading of nucleotides. And this is a very quick technology. And more, this is also an accurate technology that can be used in a clinical context to do long read sequencing. And using long read sequencing, we can tackle problems such as the complex alleles, the haplotypes that also may be implicated in disease, and this can be solved by this technology. Optical genome mapping is also a single molecule technology that allows to detect cryptic and complex structural variants, especially when combined with long read sequencing. As you can see here, this is an example that was published a couple of years ago, where a complex structural variant that remained undetected for several decades was identified using a combination of optical genome mapping and long read sequencing. So this is, I will conclude with, this is my one but last slide. I think it's important to do optimal genetic testing in the clinic to have an early genetic diagnosis, because we know that this is one of the conditions to be eligible for gene therapy. So can anything improved in genetic testing? Yes, we have now a good mainstream testing in the clinic that is pretty accessible in Europe. Mostly this is exome-based and more also genome-based. This is implemented as a standard of care. What are emerging approaches? We have multi-omics with RNA-seq that is already implemented in the clinic. We have the single molecule technologies, long read sequencing combined or not with optical genome mapping. And then very important is that we should work in a multidisciplinary context with clinical and genetic experts on board. And we have to be aware of the clinical validity and the limitations of the tests. And with this, I would like to end and to thank you. And I would also like to thank the teams, fantastic teams we are working with, also especially ERN-EYE, the Transversal Working Group 6, RedVar, ERDC, and the clinical colleagues and patients who have confidence in our expertise. Thank you.
Elfride, thank you very much for this talk from the point of the medical genetist, completing the talks of the clinicians. So thank you very much. And in the end, as the fourth presentation, I'm very happy that someone who rather represents the voice of the patients agreed to talk here also to us. This is Mr. Lacerda, dialed in from Berlin, who is the father of a patient, of a young boy having treated with Luxturna, and we are happy to hear your experiences. So please, the floor is yours. Thank you very much.
Okay, thank you for inviting me. So yeah, today I'm here like to talk a little bit what happening with me, with my family, because it was not with me. It was like the disease is not on me, it's on my son. So I want to share it a little bit. So now my son, he is 7 years old. So we discovered it like 3-4 years ago. He was 4 years old when we discovered it. But since he was born, we noticed that he had some difficulty to see at night. So I could see, everybody could see, but he could not see. And for us, it was like, okay, but okay, he is still before like 1-2 years, so 1 year old, 2 years old, he was still young. So he was thinking, no, his vision is still on development. It should be something natural. But then when he was 3 almost 4 years old. We had the second son. With the second son, when he did like 1 year, so we could compare both kids. And then now we see that he has some problem. It was clear for us. So we started investigating it. So we went to one ophthalmology, the second, the third, and it was a little bit too hard to discover the issue because it's very rare, as we know. But with lucky, we in good hands. So someone that opened our eyes and say, hey, you should investigate it because it is something a symptom of retinitis pigmentosa. Maybe he can have a congenital disease. So we did the genetic mapping of the retina, and then the results were positive. So on this day, like for us as a parent, as the father, the mother, like our words almost ends because, oh my God, now what should we do? Why it's happening with us? So in the beginning, it was real hard, but like we knowing that situation of our son, we started investigating how to save him because, okay, now the purpose of my life has changed. Now I want to save him because I don't want. I know that he can survive without seeing, being blind, but it's not the ideal world. The level, the life will be more hard if it has this condition. So we start investigating. Then at the same time, we were lucky because I received a job proposal to move to Germany. So we are on that transition on moving to Germany, but we are discovered his situation back in Brazil. And when we moved here, I keep searching for the treatment, and then I could find, thanks God for Katarina Stingl, Dr. Katarina Stingl. And then we started the treatment, the process. So yes, it was real hard in the beginning, but before the treatment and before the day also before the eye surgery, like we are very apprehensive because we know like something bad could happen during the surgery. And since it was the first eye, for us as parents, it was the most concerned one because if something went wrong in the first one, we will be very unsure what to do next because now we only have one eye to treat. But luckily, it was real good. And the second day, he did the treatment on both eyes. Everything was perfect. Everything was good. He had a very good recovering. Nowadays, for us as a family, we can realize improvements on him because before he could not like walk by himself at night. He should be always holding his hand because he could not see. And nowadays he can walk by himself. He can, not in completely dark for sure, but he like with low light, he can see much better now. He can also ride his bike, moving on the way back from the school. This is something very good. One thing that I would like to share, because I know that because my son was a kid, and I know the treatment is very painful for a kid to pass through it, and also the post-surgery, he has to do like to be open the eyes, to put the drops, and it hurts a lot. But one thing that we did as a parent, and I think it's helped a lot for him to accept the treatment, to be collaborative. It was we explaining him everything. We said, hey, Henrique, we did it because talking with some parents, they said, are you hiding this? Or he knows. No, we never hide it from him. So we explaining everything. We say, Henrique, you have this disease. We need to treat you. You have to pass through this process. It not be easy. You are going to suffer, but if you don't do it, you can get blind. What do you prefer? He said, no, I don't care. I don't want to get blind, Daddy. So knowing this, like he cooperated a lot. He couldn't, even suffering, he could open the eyes to put the drops. And this is something that I recommend for all the parents that has a son or daughter in that situation, to explain him before the surgery, make him know that this is very important for his life. Yes, in the other case, again, me and the family is very happy with the after surgery. Yeah, that's all. Thank you.
Yes, so thank you very much for this excellent end of our presentations. This was the patient perspectives, which of course matters the most. So then, thank you all speakers today. Thank you very much for sharing this excellent knowledge with the audience in the ERN-EYE community. And I would like to wish everyone a very nice evening, and looking forward to see you in some of the future ERN-EYE webinars. Goodbye and nice evening to all of you.
Thank you and bye-bye.
Well, bye-bye.